chat, and we might be able to get to them before the end of the session. Well, thank you very much, Rachel. That was fantastic. We'll move on to the last speaker of this session, which is Natalie Groves from Public Health England, now called the UK Health Security Agency. And Natalie is a senior bioinformatician there working on the COVID-19 response in the genomics cell. So Natalie, are you able to share your screen and do your presentation?
Yeah, can you hear me okay?
Can hear you, can't
Great.
see you.
That's okay.
Okay.
That was my plan.
All right, that's fine. But can see your screen now.
Okay, great. I've got some new headphones, so I was just checking that you can actually hear me on Zoom.
Looks all good.
Great. Okay, so I'm just going to kind of go through some of the kind of processes that we've set up within the genomic cell as part of the COVID-19 response, and particularly looking at how we are detecting and defining variants. And then at the end, I have some kind of case study type examples of, things that we have to do to kind of continually refine the variant definitions. So hopefully it will be interesting for all of you. So the genomic cell was set up in late 2020 to interpret the genomes being produced by COG-UK and help with the national public health response and respond to regional and local queries. So, I mean, obviously at the time we had a much smaller number of genomes, and we were kind of helping with molecular epidemiology questions and dealing with kind of small outbreaks in specific settings to help with those kind of local responses. Now our response is much different because of the case numbers and things like that. But we are a relatively small team of bioinformaticians. It has grown recently, but we did start off only being 3 people. Now we're up to, I think, 8, which is really nice to have some extra bodies in the team. But we also have some virologists, clinicians, epidemiologists, modeling staff, and general data scientists within the genomic cell as well. And we have a team of support staff that kind of deal with all of our admin woes. We have 3 main areas for bioinformatics. There's a studies and surveillance team who deal mostly with the kind of outbreak reporting or helping with some of the studies, so the reinfection studies or vaccination studies. There's a development team, which is the team that I lead, and we deal with more kind of the setup of the processing scripts and sort of investigating and developing the processes for QC and variant definitions and line lists and all of those kind of things. And then the horizon scanning team, that are kind of their job is to kind of look for those upcoming variants and make sure that we can spot them before we have a real issue on our hands. At the moment, we're still operating a 7 days a week service, which is obviously pretty tough when we've got a limited number of people, especially in the senior team. But hopefully that will start to kind of turn down in the coming weeks. We also have a role in responding to press queries, queries from international partners, parliamentary questions, and also public freedom of information requests when those relate to genomics. And we work with all of the other COVID-19 response cells within the organisation and with academic partners, those that were involved in COG-UK, etc. We obviously have a range of functions across the teams, but we have lots of kind of regular reporting tasks that we need to make sure are completed, and some of that information is then made publicly available in things like dashboards or the technical briefings, which some of you may already be familiar with. We are a very small part of the National Variant Assessment System, so there are lots of different pieces that that feed into eventually declaring a VUI or a VOC, but we are quite often the kind of starting point at the beginning. So we are scanning for these initial signals, looking at their growth, any kind of signals of immune escape or severity based on, you know, the genomic data, and then the epidemiological data that we're able to associate with that. And we have these kind of early signals that we monitor for changes in those kind of things. Then we would ask for kind of some initial studies to be done around those signals so that we can get a better understanding of actually phenotypically what's happening, and whether there is actually a growth advantage or any kind of concerns around the kind of pseudovirus neutralization and things like that. And that's the point where we would consider declaring a VUI. And then for something to move to a VOC, it needs to have a much more robust analysis, including where possible the live virus studies. And so that's kind of another level of evidence that's required for us to kind of declare at that higher standing, because that comes with a lot more kind of public health action and has a bigger impact on policies. So we have to be kind of a bit more sure about what's going on before we can do those kind of things, because of all of the downstream implications of declaring that. So I'm going to kind of talk a little bit about the horizon scanning process, because that kind of is really crucial to our detecting of the variants. And we have kind of 4 main data feeds that we have kind of routine reporting for. So the first one is cluster detection, and that is using kind of the phylogenetic trees and estimating growth rates to identify clusters that are growing faster than their kind of equivalents in that region, compared to kind of other variants. And this also applies to kind of additional mutations on top of an existing variant. So if you see a particular cluster within a variant that's growing faster and it has very unique characteristics, those are kind of the points at which we declare those kind of second level variants, which we have seen a few of over the course of the pandemic. The next piece is the mutation scanning. So we look, this is slightly out of date actually, I should have updated it, but the mutation scan is now kind of running in a more agnostic way. So initially we were looking at kind of known antigenic and avidity mutations, but that kind of posed a few problems when we had things like Delta that actually didn't really tick any of the boxes, the existing boxes that we had. So never really flagged on that side of things. So we kind of expanded what we're looking at, and it's kind of a big hurdle for us to get over, because there's lots of things that we don't necessarily know much about, but it is a much better way of picking out things that really are emerging and could be a cause for concern without any kind of preconceptions about what the risk would be for those particular combinations of mutations. We also take all of the data from GISAID, so we look for emerging lineages or specific mutation constellations that look like they're growing in a particular region, and we obviously look at the existing variants appearing in new countries, because that has an impact on the kind of border assessments, and you know, when we go back to having red, amber, green lists, if we see these kinds of patterns emerging. We also have a specific look at the imported cases. So this is taking the UK data from travel associated cases, and that kind of helps us to just support what we're seeing in GISAID, or kind of identify things that we're not able to see in GISAID because there's lower sequencing capacity in those countries, or they're not very quick at uploading their data. So it kind of gives us a more controllable data set that, you know, we know the inputs and outputs and the limitations of, because it's kind of within the UK data. And then we have lots of other things that kind of feed in. You know, we get we pick things up on Twitter like everybody else. And we have various other partners that are looking at things, and they might pick something up that hasn't quite reached a level where it will hit the thresholds for our formal reporting. So we get those kind of early warning signals from some of our academic partners as well. So thinking about what a new signal actually is, we might see a new constellation of mutations that may or may not have known antigenic or avidity mutations. And those might, be emerging, and, we'll have a couple of clusters, or we've seen them imported several times, or they are overrepresented in vaccinated individuals. Those would be kind of warning signs for us. Also looking for increase in frequency of just a specific mutation, and that would be additional to a variant or in something new, that we would kind of pick up those signals. Again, the mutation constellations within a specific group of individuals, or within kind of a specific setting. So perhaps there's an age-related issue, if we're seeing it mostly emerging in children or in kind of care homes, etc. And then obviously looking at the clusters with high growth rates. So just to kind of illustrate how this kind of works, I have this little diagram. So the initial signal is this kind of cloud of things that we're aware of but aren't really monitoring, or we don't feel is of a particular concern that we need to take action, or add resource to investigating those kind of things, but we do know about them. Then we have signals in monitoring. So that's something where we have detected this signal that we think actually may be of concern. So we're going to watch it closely and see how it develops over the next few weeks, to just make sure that we actually don't feel like it is of a major concern, and that we can either it then gets escalated if it is of concern, or it will be downgraded and removed from monitoring if we think actually it's not of concern. Then obviously we go into our VUIs and VOCs, and that has a specific threshold, as I talked about before. So when we do decide that we have a new variant, obviously we need to define that so that we can detect it as a on a kind of ongoing basis as part of our routine reporting. And there are kind of various things that you need to consider when you're defining a variant, and I'm going to kind of go through a few of those in the next few slides. But the kind of first thing is kind of really nailing down your data set that you're going to say, this is my variant, this is what my definition needs to detect. So I have kind of two examples here where, so on the left, we have in the yellow our variant sequences. And so you can see it's a very small number of sequences. There's some quite long branches there. And some of that is kind of data quality issues rather than kind of genuine variation across this variant. And so we have to be mindful of that when we're trying to produce these definitions, to make sure that we're not discounting things or including things that are just, victims of quality. And then on the right hand side, this is a much larger set of sequences. So this particular variant was circulating, and we'd been aware of it for a long time. It was circulating in other countries, but it took quite a long time for it to become of concern in the UK. So we didn't declare it as a variant until it had been around for quite some time. But you can see in the tree on the right, there's a section just in red of the tree. So these are all things that are called as this lineage by pangolin, but when you put them in a tree, they're very obvious outliers. So there's clearly some issues in terms of the mutations in there, and so those ones, when we're creating a definition, would be excluded because they're going to reduce the number of sequences where you see a viable defining mutation, or they might introduce some new mutations that actually are not variant defining, they're kind of systematic errors or just general poor quality regions. So yeah, defining your kind of sequences of interest is really important when you are detecting a new variant, and I'll kind of show you an example of that later when we come to the revision of the definitions. So when you're kind of picking mutations that are going to go into your definition, you obviously want to make sure that you're picking mutations that are very representative of your clade, but are as specific as you can be to the clade that you're interested in. So in this image, you have each row as a sequence, and at the top of that is the SARS-CoV-2 reference. And so the top set of sequences are the ones actually in my clade of interest. So you can see there are 3 sort of bases across this region that look specific to this clade, but there are some that are missing it. So it's really important that in those cases you make a proper assessment of whether that's a data quality issue, or actually if in the part of that variant that you happen to have sampled in your sequencing, you are picking up things that have acquired these mutations that aren't actually ancestral to what you're defining as your variant clade. But the 2 mutations at the very edge of this kind of piece of the sequence are present in a much larger group of sequences. So you see this mutation outside of the clade, and that means that they're not going to be very specific. So you'll get a lot of hits to those mutations in your sequence data because they're maybe ancestral B lineage variants, or they're the D614G that Rachel was talking about earlier. So you get lots of hits, and it makes it more complicated trying to filter out the things that look similar but aren't the same. So you want to try and increase your specificity as much as you can. Once you've defined your mutations, you then need to set your thresholds. So we have 3 kind of groups for the definition. We have a confirmed definition, and at the moment, we generally require all mutations unless there are an abundance of mutations, and a lower combination of those is always unique. So the first example of that for us is Omicron, where there were so many mutations in those sequences that if you used all of them in your definition, you didn't need to have all of them present to confirm that it was Omicron. And that was particularly impacted by amplicon dropouts in the Arctic primers. Currently, we don't allow wild type calls in most of our definitions. Again, an exception to that is Omicron, because there are so many mutations and we don't necessarily understand what Omicron looks like ancestrally. We allow a small number of wild type mutations to try and help bring in any of those ancestral sequences that we didn't have at the point of writing our definition. And then we have a probable definition. So this is where you're kind of trying to strike a balance between sensitivity and specificity, because you need to make sure that the number of mutations you require is specific to your variant of interest, and you're not going to get calls outside of the specific clade. And that's particularly important when you have multiple variants within the same or very closely related variants. So for example, the 3 variants that were declared from the 617 lineage, one of which was obviously Delta, those still share a lot of ancestral mutations because they diverged, fairly recently. And so you have to kind of strike that balance so that you're not calling all 3 of these variants on the same sequence. You need to kind of make sure that you're sensitive enough, but also you don't want to start excluding things that have quality issues, and actually you're just missing pieces of the genome because you have amplicon dropout where you're using high CT samples and things like that. Our low QC definitions are set to mutations required 0 because we need to capture sequences where we can't exclude that variant, so that if we're reporting that onwards to health protection colleagues, they know that although we can't confirm that it is or is likely to be this variant, we also cannot rule it out. So if they have a cluster of a specific variant and this specific case forms part of that cluster epidemiologically, they know that they can say, well, the epi links are strong, so we're going to say that this is part of the cluster because the genome sequencing has been inconclusive, rather than us saying it's not that variant when actually there is a chance that it could be. All of these definitions are on GitHub for anyone to use. So if you want to have a look at those, we publish these YAML files on the GitHub with all of the mutations and the categories and thresholds that we are using as part of our reporting. So feel free to go and have a look at those and use them should you wish to. And so the next thing, once we've defined a variant, is actually to review the definition. So this example is the B11318 lineage, which was one of the variants we declared last February. And partway through last year, we noticed that we were seeing things that were classified by pangolin as a B11318 sublineage, but were not being called by our mutation definition. And so we were missing these cases. When I looked at the tree and the mutations that were present, it was apparent that there were 2 mutations that were missing in this subset of sequences. And that possibly is because when we originally defined this variant, we had, I think, maybe 7 or 8 sequences, and 5 of those were in a single cluster, a household cluster. So we really didn't have an understanding of the variation that was present in this particular lineage, and were defining the variant based on this kind of the clade at the top of this tree. And then we started sampling in our imported cases more ancestral sequences from the region that the variant was actually originally circulating. So we then removed those mutations from the definition and were then able to call a much wider set of the sequences that we were seeing. Then there's also an issue with increasing sensitivity. So initially, when we declared Delta, we set a variant definition that had 12 mutations, and a large number of those were shared with the other 617 clades. And so that meant that we had to have a very high threshold for our probable definition to make sure that it was specific to the B1617.2 clade rather than the 0.1 or 0.3 clades. So when we kind of did this revision, we looked at which positions were dropping out quite often in the sequence data because of problems with amplicons, or just generally those regions are more difficult to sequence. And we found that actually there were some positions that we could remove that weren't kind of unique to the B1617.2 clade. So they weren't necessarily informative positions in terms of this definition, but they caused a lot of problems in terms of dropout. So once we removed those, we could lower our probable threshold to only requiring 4 mutations to make sure that we had a variant specific combination. And that meant that we had a lot fewer falling into that low quality category, and were able to more accurately and more sensitively detect this particular variant. And then the last example I'm going to give you is an Omicron example. So initially, when we defined Omicron, it was based on what has now become the BA1 lineage. And then we subsequently had BA2 and BA3 designated. And that meant that we had to refine the definition to make sure that what we were using was specific to the lineage that we were trying to identify. And so initially, when we defined this variant, we obviously took as many of the mutations as we could, but these two closely related lineages obviously shared some of those B11529 sort of ancestral mutations, and we needed to remove some of those to make sure that we were still detecting the right lineage. And that required us to identify a set of kind of clade specific mutations or combinations, and then make an assessment of the sensitivity and specificity of the definitions we were using. So as an example, this image just shows you the mutations that were present in spike for BA1 and BA2. And the kind of color of the block shows you the proportion of sequences that contained this mutation. So you can see at this point, we were already starting to see some diversity in BA1, and we obviously needed to make sure that we weren't using these mutations that weren't present in all of the BA1 sequences. So we wanted to avoid those in our definition. But you can also see that there's quite a lot of overlap with BA2. So a lot of those mutations we then couldn't use because it meant that our definition wasn't specific enough across the BA1 lineage. So we came up with these new definitions. I'm not going to dwell too much on what the mutations are, but the point I kind of wanted to make here is that we haven't just used clade specific or lineage specific mutations in these definitions. We have included a subset of mutations that are shared between both, but that we felt were fairly unique to the B11529 clade. So that meant that if you had sequences that were slightly poorer quality, you had a better chance of picking up at least Omicron mutations. So you could say, I have enough evidence that this is within the B11529 clade, and then I have some evidence that it's either BA1 or BA2, and you didn't need quite such a high number of mutations that were specific enough to be able to call these variants at least to a probable threshold. So the last thing I have is just some basic do's and don'ts for defining a new variant. There are obviously lots of things you need to consider, but these for me are kind of the main things that you need to kind of think about. So as I said, you do really need to clearly define your clade of interest. And it's also really helpful to make sure that you have some ancestral data, or at least non-variant data, so that you can pick out those mutations that actually are not specific to your lineage of interest. It's really important that you consider synonymous changes, because actually sometimes those provide a kind of higher specificity than some of the non-synonymous changes, particularly those around kind of functional sites. So in the RBD across spike and things like that. You do need to make sure that you're checking for false positives when you're defining your clade, and also when you are testing the definition that you've come up with. And then it's really important that over time you refresh and refine that definition to make sure that you're capturing everything. In terms of don'ts, the quality of the genomes you're looking at are really, really important. You need to make sure that you're not excluding mutations because you have dropout in some of your sequences, or you're not picking mutations that clearly have some kind of systematic issue with detecting them. So that's going to drag down your sensitivity for this particular variant. I generally advise that you don't include indels in this kind of definition, just because they're so much more difficult to deal with bioinformatically, in terms of making sure they're aligned properly, that they are actually called correctly, dealing with when they're in the faster sequences ends compared to dashes. All of that kind of stuff just makes it much more complicated. So if you don't need them, just don't use them. And then thinking about your thresholds is really important, and making sure that you set those appropriately for, you know, the acceptable dropout that you can deal with. You know, at the very beginning of declaring a variant, it's really important that you have absolute sensitivity. And, you know, if you have to sacrifice some of your specificity at that point, you have to make that judgment call. But if you're trying to, you know, stop a variant from taking hold in your population, you really need to maximize your detection sensitivity. But if you're trying to differentiate between two things, then your specificity is obviously more important in those cases. So you really need to decide what it is that you want out of the definition and make sure that the thresholds you set are appropriate for those aims. And the last thing is just to say that we wouldn't be able to do any of this without the huge number of sequences available and all the people that work really hard to produce those and process them for us. And a lot of processes and data feed into declaring and defining variances, not entirely on us. Shout out to the technical briefings, which I think are a really nice resource for people to look at the kind of analysis that we're currently undertaking in terms of variance. And then, acknowledgements to a huge number of people in UKHSA and outside. But yeah, happy to take any questions.
Thank you very much indeed. It's a really informative talk about how it all works, certainly from the UK side, and hopefully that was very useful for everyone. We do have a couple of questions, and it'd be great to see some more questions in the chat. Li Ching's asking, thanks for your presentation. There are some mutation signals, some are not, for example, recombination signals. So I guess the question is, how do you distinguish between recombination and mutations, I think?
I mean, that's a really good question, and one that we haven't really nailed down yet, I don't think. I mean, we try to take, advice from other people as well. So, we're not solely working on this on our own, and there are lots of other people that will kind of also make those assessments, and we try to take as much data in as we can. But I think, we try to make that assessment based on kind of what we know is circulating in certain regions, whether they would have the opportunity in that particular environment to recombine, to know, if there is a likelihood that that did actually happen. And just, with recombination, looking for those kind of breakpoints to pick those things out, I think is, really important. And just kind of making that judgment call in terms of, do we think that actually there's a functional reason why it would independently develop this combination of mutations? Do we know that it's a really successful combination, or do we know that this set of mutations confers a particular advantage, and actually seeing those arising independently wouldn't be that much of a surprise? I think, that's a really important question to ask around that kind of specific topic, but it is a really hard thing to differentiate, and I'm not sure we've necessarily nailed it just yet.
Okay, great. I can't see. I mean, there's a question about is neighbor joining still used for large phylogenies? I feel like that's a question, a general question, not necessarily relating to your talk, but you can answer it. Is neighbor joining still used for large phylogenies?
I mean, generally we try to decide on tools based on how quickly we need information, yeah. And the resources available to us. So a lot of times it's, a toss up between speed and accuracy. And so sometimes if we just need a really quick look over a phylogeny that we can make a best guess, then we potentially would put it through neighbor joining. But where we're trying to make these kind of more formal assessments of things, then we would be more likely to try and get a maximum likelihood tree together, or at least best equivalent. Yeah,
I mean, neighbor joining isn't entirely inappropriate for a quick tree, particularly with SARS-CoV-2, I think, yeah. Because it's a relatively low rate of homoplasies and recombination that would kind of mess that up. But I guess probably most of the time you'd probably just go for an ML method because they're not that slow if there's small numbers of genomes. Brilliant. All right. Well.